So this is the second half of Chapter 3, pages 52 to 63 if you're keeping score in your textbook. This is part 2 of the Basic Principles of Heredity lecture. I guess you're a glutton for punishment if you've come back for more, but actually this is going to take what we've learned in the last lecture and really make it special. What we're going to talk about today first is probability. Yeah, basic mathematical probability. The genetics problems we'll be reviewing in this lecture are going to be made remarkably more easy by using the general principles of probability, and so I don't want to just assume that everybody's comfortable with that kind of math. We'll go over that in its very basic sense first before we go anywhere near genetics today. But then once we feel comfortable with probability, we will be using those skills and those tools to predict the outcomes of genetic crosses. We'll review what we mean by test crosses, because we're going to be using test crosses more and more as we continue on with Mendelian genetics, and then we will get to dihybrid crosses. If monohybrid crosses were crosses that involved one gene, then dihybrid crosses are crosses that involve two genes simultaneously. We'll end the lecture with a very brief introduction of the chi-square tests from statistics. This is a goodness of fit test. We are not really going to use this math directly in this course, but any course in genetics wouldn't be complete without a full discussion on chi-squared, and so we'll take care of that now, just so everyone's familiar with it, everyone has heard of it, and you wouldn't be shocked if you were to see it again, not necessarily on one of my exams, but things like the MCAT, the pre-vet exam, the pre-dental exam, etc. All of those are very likely to include questions on chi-squared. But anyway, so Punnett squares. We talked about Punnett squares in our last lecture, and Punnett squares are great. I'm not going to knock Punnett squares. I appreciate their value. They're easy to understand. They're easy to use. They visualize what's going on in the genetic cross. They let you keep the gametes straight and keep your thinking straight. So Punnett squares are awesome. But Punnett squares really are only awesome for single-gene monohybrid crosses. After that, even at the dihybrid level, Punnett squares get really confusing really fast. And so I don't even want to go down that road. Most phenotypes, most traits, most things that organisms look like or have are actually the result of many different genes working together to cause that trait. And we're going to touch on this at the very, very end of our semester when we talk about a field of genetics called quantitative genetics. But just suffice it to say now, most traits are the result of many genes. So when we're looking at single traits in more advanced organisms and more advanced traits, it becomes important to be able to predict the inheritance of multiple genes in a single cross. Most traits aren't single gene traits like tongue curling and pea plant height. Most traits are multi-gene traits, and so we got to keep track of multiple genes at one time. Punnett squares are quite difficult to use when you're talking about multiple genes, but we need to be able to track and predict the inheritance of multiple genes. So how do we do this if we can't use Punnett squares? And the answer is by using very simple principles of mathematical probability. It's unfortunate, but for the most part, the United States educational system is in shambles. It's in a pretty bad state. Many of you are Massachusetts residents. Massachusetts has one of the best public school systems in the country, so you might be in a better position than most, but I don't want to assume anything. It's quite possible that many of you were not adequately taught probability in high school, or even if it was adequately taught, you're not comfortable with it. And so I don't think it's fair to just jump into dihybrid crosses assuming everybody's fine with probability. I'd like to take just a few minutes to review the basic laws of probability first. If this is review for you, then I apologize for wasting your time, but it never hurts to review. If you're a probability whiz, you may want to just fast forward the lecture a couple minutes and pick up where we go back to genetics. But again, I think there's value in going over this quickly right here. So, in its most basic sense, a probability is simply a prediction. It's a mathematical, quantitative prediction. Probability is a quantitative way, meaning it uses hard and calculatable numbers, to determine how likely it is that something's going to happen in the future. That is the prediction. So, as its mathematical definition, a probability is the number of times an event happens over or divided by the number of opportunities that event has in which to happen. Pretty confusing definition. A real-world example makes much more sense. You flip a coin. What is the total number of opportunities for something to happen when you flip a coin? There's two. There's two options. There's two possible outcomes, a head or a tail. What's the probability of flipping a head? Well, now we're saying, what's the probability of doing one of those two things? What's the probability of getting a head when there's one head, 1, over or divided by the probability of total things that there are, well, 2, a head or a tail. So the probability of flipping a head on a coin is 1 in 2. On a die, probability of rolling a 5 is 1 in 6. Again, how many 5s are on a single die? There's 1. How many numbers or possible events are on a single die? Well, 6. So 1 in 6 is our probability. Probabilities usually aren't represented as these types of sentences, 1 in 2, 1 in 6. They're usually represented as a fraction or a decimal, represented on a scale from 0 to 1. So truly, the probability of flipping a head is 0.5, 1/2. The probability of rolling a 5 is 0.166666666, or rounded to 0.167. So we fall on that range from 0 to 1. If a probability equals 0, it means mathematically that the event will never occur. Never, ever occur. The probability that I will live forever is 0. It's not going to happen, no matter what. And then on the other side of the scale, a probability of 1 means that the event will definitely occur. I hate to be morbid or, morose, but it's true. What's the probability I'm going to die someday, at some point in this life? It's 1. I'm going to die. The closer to 0 we are on that range, the worse the chances are. In other words, the less likely the event will happen. And the closer to 1 on that scale, the better the chances are, or the more likely that that thing will happen. So I'm sure all of you are familiar with that, but that's the basis of probability. So like any good mathematical field, probability has its own laws. There are only 3 laws of probability, so you don't need to freak out too much. And I've already taught you one without you even realizing it. I've taught you a law of probability. The first law of probability is that probabilities range from 0 to 1, with 0 being not happening and 1 being definitely going to happen. The second law of probability is that some events are mutually exclusive. What do we mean by that? Well, we mean that when events are mutually exclusive, when one event occurs, it automatically invalidates all the other events and makes them impossible. Again, let's go back to that coin flip. If I flip a coin, and I flip that coin one time, and I flipped a head, it is physically impossible for me to also get a tail too on that flip. That's the key. One event, a coin flip. If I flip a head, I can't get a tail on that flip. They are mutually exclusive. Once I got one outcome, I can't get any of the others. Roll a die, you roll a 2. You can't, in that roll, have gotten a 1, 3, 4, 5, or 6 if you've already gotten the 2. They're mutually exclusive. When events are mutually exclusive, that means you can take their probabilities of all the individual events and add them up, and you will get 1. It's called the sum rule. So coin flips are mutually exclusive. What's the probability of getting a head? Half. What's the probability of getting a tail? Half. Is flipping a head or a tail mutually exclusive? Absolutely. You make the other outcomes impossible. Are there any other possible things that you can get when you flip a coin? And don't anyone say it lands on its edge. Come on now. There aren't. So again, what's the probability of getting the head? 0.5. What's the probability of getting the tail? 0.5. Are they mutually exclusive? Yes. Are there any other possibilities? No. Add up 0.5 and 0.5 and you get 1. Perfect. You can do the same with a die roll. It doesn't add up as cleanly because we have to round to 0.6 or 0.7, but the message is the same. The probability of rolling a 1 plus a 2 plus a 3 plus a 4 plus a 5 plus a 6 is 1, because those are mutually exclusive events. We can play some tricks with this math. Because the probability of flipping anything is 1, we can manipulate that math and calculate the probability of flipping a head as 1, the probability of flipping at all, minus the probability of flipping a tail. Why does that work? Well, think of it in real, just English, as a sentence rather than math. I tell you, I flipped a coin, and I did not flip a tail. That's what that math is saying. I flipped a coin with a probability of 1, and I did not get the tail. Well, you know right away, if I didn't get the tail, I must have gotten the head. But what you're really doing is that 1 minus 0.5 for the tail equals the 0.5 for the head. And so that's the idea of mutual exclusion. Again, this is called the sum rule, and the sum rule is called the sum rule because based on the sum rule, you can add individual events and get a combined probability. Try to think of something that's mutually exclusive in genetics. I'll give you a second or two. Have we talked about anything that kind of matches that coin flip example? Anything that's mutually exclusive? If you came up with the right answer, you're well on your way to an A in the course. The mutual exclusion that we've talked about so far is which of the two alleles a parent gives to a particular offspring. You remember in our last few lectures, both when we talked about meiosis and also when we did the first half of basic heredity here, Mendelian rules, we said that every parent has 2 alleles. But only one of those alleles can be given to a gamete, and only one of those alleles is contributed to a single individual offspring. That's a mutual exclusion. Let's say I'm heterozygotic for A. I have one big A allele and one little a allele. I'm heterozygotic. I can give the big A allele or the little a allele to my daughter, but I can't give both. And more importantly, once I've given the big A allele, I cannot physically give the little a allele too. Just like once I flipped a head, I can't also flip a tail. So which allele is donated during reproduction, which allele makes it into a specific gamete, is mutually exclusive. The third law of probability states that the probability of 2 independent events occurring independently but together is equal to the product, the multiplication product of their individual probabilities. This is called the product rule. Think of the lottery. The lottery provides a perfect example. I'm not sure how well this is going to come through on Integrity, but I'm going to give it a try anyway. I was 9 years old when this lottery was conducted, actually just a couple short months away from turning 10, and this lottery has nothing to do with me. I was never in Illinois, but this is an example of some of the things that would be on TV when I was a kid. I would actually try to predict the numbers. Right before the balls popped up, I would say numbers and try to see if I had any ESP, any ability to see the future 3 seconds in advance. But we're going to use this as an example for probability.
WGN brings you the official winning numbers of the Illinois State Lottery's daily game and Pick 4. Tonight's drawing is supervised by Jerry Simon, partner with the accounting firm of Laventhal and Horwath. Here's Ann Wendell with tonight's 3 daily game machines in a randomly selected order. Each has 10 balls, 0 through 9. Drawing starts by mixing the balls in the first machine. Next, the balls in the second machine. And then the third machine. Now here is tonight's first winning daily game digit. 0. Tonight's second winning digit. 9. And the third and last digit. 2. So for tonight, the official daily game winning number is 092. Now the Pick 4 machines in a randomly selected order. The balls are mixed in the first machine. Then the second machine. Then the third machine. And finally the fourth machine. Now the first winning digit for Pick 4. 2. The second number. 1. The third number. 2. And the fourth and last digit is 1. So for tonight, the official Pick 4 number is 2121. Join us again.
So which number is picked is random chance, but still probability can predict for us the chance of each winning lottery combination. Each number coming up is its own independent event, and that's the first part of the product rule. Events have to be independent. In other words, what ball came up in the first canister has no impact whatsoever on what ball is going to come up in the second canister. Each ball coming up in each of those canisters is completely independent. So let's consider the Pick 3 first. The first number to come up in the Pick 3 was a 0. There were 10 balls in that canister, 0 and then 1 through 9. So the probability that the ball that came up was going to be that 0 was 1/10, 1 in 10, or 0.1 in a fraction, in a decimal. The second ball to come up was a 9. Now, the 0 coming up in the first canister had no impact whatsoever in what was going to come up in the second. So we treat it as an independent event, and the probability of getting that 9 is also 1 in 10, or 0.1. And the third number to come up was a 2, and it had a probability of 1 in 10 as well. But these are independent events, and so the product rule says the probability of getting all three of those independent events together, in other words, the probability of getting the 0, the 9, and the 2, is the same as the product of their individual probabilities. So 0.1 times 0.1 times 0.1 is 0.001, or 1 in 1,000. So the probability of getting 092 in that pick 3 lotto was 1 in 1,000. We can do the pick 4 as well. The pick 4 number was 2121, but each of those numbers was an independent event. The probability of getting the first 2 was 1 in 10. The probability of getting the first 1 was 1 in 10. The probability of getting the second 2 was 1 in 10. The probability of getting the second 1 was 1 in 10. The probability of getting the combination 2121 is the same as the product of all those independent probabilities. 0.1 times 0.1 times 0.1 times 0.1, or 0.1 to the fourth power, which is 0.0001, or 1 in 10,000. So the probability of getting that combination in the pick 4, 2121, was 1 in 10,000. So as you can see, the number of possible winners, or in other words, the chance of getting the right number, goes down. It gets harder with each additional event because we're multiplying in a new probability. I like to think of these sum and product rules as sentences, as English sentences. It makes the most sense to me that way. And the one word, the English word that best represents the sum rule is the word or. And in my mind, the word and best represents the product rule. I'll show you what I mean. What's the probability of flipping a coin one time and getting a head or a tail? Think of that for a moment. I flip a coin once and I'm asking you, what's the probability I get a head or a tail? Hopefully you said 100%, or a probability of 1. If you flip a coin, you're going to get a head or a tail. What's the probability of flipping a coin 2 times and getting a head and then another head? Well, now I have to think of the probability for the first flip is a half, and then the probability of the second flip is a half. So the probability of flipping a head once and then flipping a head again is a half times a half, the product rule, or 1/4. Both of these rules come into play for genetics. We already said it once, but the probabilities of individual alleles at individual genes is governed by the sum rule. They're mutually exclusive. Either you have a dominant A or a recessive A at a particular locus. Or, thinking about it a different way, when your mom contributed some genetic information to you, she could have given you the A allele or the little a allele, not both. The probabilities of particular genotypes within our own genomes is governed by the product rule. For each individual allele, we have 2 of them, and each of those 2 alleles is an independent event in our genome. So if I have the genotype big A, little a, that's the probability of having gotten a big A allele from one of my parents and, separate event, the little a allele from my other parent. So we multiply those probabilities. These are the products of the individual inheritance patterns for those traits. This figure's from your textbook, from your genetics textbook, but it just breaks this down in figures. I think it might be helpful. The product rule, or the multiplication rule, says that if you roll a die and you roll a 4, the probability of getting that 4 is 1 in 6. If you roll the die a second time, and you roll a 4 again, the probability of rolling 2 4s in a row, or 2 separate events, 1/6 for the first roll times 1/6 for the second roll, is 1/36, the product of those 2 fractions. The sum rule says, well, let's say you roll a 3. What's the probability of rolling a 3? 1/6. Let's say you roll a 4. What's the probability of rolling a 4? 1/6. But now the question is, what is the probability of rolling a 3 or a 4? Remember, the or word is the sum rule. A 3 or a 4 is 1/6 plus 1/6, or 2/6. So the probability gets easier as you add in OR events. The probability gets harder as you multiply in AND events. Hopefully you're not lost. Hopefully this was some review and makes some sense. If you are lost, it's okay. We're going to use these rules now for genetics. So as opposed to the last time I thought you might be lost, where I said back it up and watch it again if you have a chance, don't bother doing that this time. If you can understand the probability in the context of genetics, you'll be fine. This is more just to give you a good foundation to build the genetics on. So take a look at the figures if you need to. Certainly come see me if you feel you need to. My door is always open. But don't freak out too much if all this math just kind of went a little bit over your head. So let's use this information. Let's give it some context in a genetics framework. So let's apply probability to some genetics crosses. I think we're all comfortable, or at least I hope, we're all comfortable with Punnett squares and monohybrid crosses right now, these single gene crosses. So before we go ahead into deeper genetics, let's use our newfound probability skills on our old, tried and true, tested monohybrid crosses. Let's consider what we worked with in the last class, a double heterozygote cross between two pea plants. This case for the height gene, we have a tall parent number 1 who's heterozygotic crossed with a tall parent number 2 who is also heterozygotic. Rather than using the Punnett square, let's see what is the probability that parent number 1 gives a gamete to the offspring that contains that dominant T allele. Well, we have two possible events, either the dominant allele or the recessive allele. And this parent's giving one of those two events, so it's the same as a coin flip. Probability is 1/2. What's the probability that parent 2 also gives her dominant T allele? Well, it's the same question. She has 2 to give, they're different, and only one of them is dominant, so it's 1/2. So what's the probability that parent 1 and parent 2 both donate the dominant T alleles to their offspring? I said and, so that's the product rule. So it's 1/2 for parent 1, 1/2 for parent 2, 1/2 times 1/2 is 1/4. So there's a 1/4 chance that the offspring is going to get a dominant T allele from each parent and be tall. Let's do the same for a dominant T, recessive T, where the dominant T comes from parent 1 and the recessive T comes from parent 2. Well, it's still 1/2 for the dominant T from parent 1. It's 1/2 for the recessive T from parent 2 because that's the other alternative the parent had, and we multiply them to give us 1/4. Let's also go for the heterozygotic offspring, but the other way around. Now parent 1 gave the recessive allele and parent 2 gave the dominant allele. Doesn't really change the math, and so we're still left with 1/4. And finally, what is the probability that each of those two parents gives their recessive allele? Still 1/2 for each parent, so still 1/4. So the individual probabilities of each possible mating event is 1/4. But that leaves us 3/4 tall and only 1/4 short. So the question becomes now, what's the probability of getting a tall plant? Well, the tall plant is the sum rule. The other way of saying that is, what's the probability of having this mating and getting the genotypes big T, or big T, or little t, big T. See, we say or now because all three of those genotypes gives you tall. So that's 1/4. When we're dealing with ors, that's 1/4 plus 1/4 plus 1/4, which is 3/4. All of this matches our Punnett square perfectly. This is what the Punnett square was doing for us, except visually instead of mathematically. 1/4 was homozygous dominant. You could think of this mating event representing this one, whereas this mating event represents this one. They're indistinguishable genotypically and phenotypically, but they are still the results of two independent mating events. And then finally, of course, we can have the homozygous recessive making up 1/4. So you get the same exact answer using Punnett squares and probability, get the same exact answer using two completely different methods, demonstrating that the probability method works. Are Punnett squares easier for monohybrid crosses? Yeah, there's no doubt about it. And you should use Punnett squares for monohybrid crosses because they're easier and more straightforward. But Punnett squares are much more difficult for dihybrid crosses and above. So we're going to use these same thinkings of probability, these same rules of probability, these same tricks of probability for the more complicated crosses we're about to do. Punnett squares are only good for seeing single matings, only good for tracking single gene events. The probability approach allows you to predict the genetic outcomes with multiple genes. We'll spend most of our time on dihybrid crosses, but you can use the probability method for matings that involve dozens of genes if you wanted to. And just to be clear, I really don't want to see Punnett squares for dihybrid crosses or above on any exams or quizzes. I'm going to put my foot down in a way and restrict you to using probability only. Even if you've taken genetics in previous courses at the high school level and you're used to using Punnett squares, you're just making life much harder and more complicated for yourself. So stick with me and try to get comfortable with this method. And of course, just like monohybrid crosses, everything we do today in the higher level crosses will allow you to predict the genotypes and phenotypes of multiple offsprings of a given mating. Just to kind of put a fine point on some of the probability laws, given all the information that's on the slide right now, what's the probability that with 3 successive mating events and 3 successive offspring baby plants, that all of them are short? We can say this another way. We can say, what's the probability of having 3 plants from a mating, and 1 plant is short, and the next plant is short, and the third plant is short? That ands should clue you in that we are dealing with the product rule. It's a quarter probability for the first plant, times a quarter probability for the second plant, times a quarter probability for the third plant. That's 1 over 64, incidentally. Now I ask you another question. What's the probability that, given 3 successive matings and 3 successive baby plants, 2 are tall and 1 is short? We could do that problem, but the answer is not 1 over 64. Because once we go to different phenotypes, and I don't give you the order, the mating event could be the first one's tall, the second one's tall, the third one's short. Could be the first one's short, the second one's tall, the third one's tall. Could be the first one's tall, the middle one's short, the third one's tall. So we have to account for each of those possible combinations. It's not as easy as the short and short and short. It's doable, we could do it, but it's not so easy. So just keep in mind that the wording of questions needs to be closely paid attention to so that you don't kind of assume any order or assume any information that's not provided in the question. We talked about test crosses or back crosses in the last lecture, but I want to review it here before we get into dihybrid crosses, because we're going to be doing a lot of test crosses from this point on. So I have an organism, we'll say it's a pea plant, doesn't really matter what it is, and I want to know its genotype at the height or tallness gene. I know the plant is tall. If it wasn't tall, I would know its genotype. If it was short, it would have a homozygous recessive phenotype, but it's not short. It's tall. So I already know a genotype it's not. It's not little t, little t. I just told you how I knew that. It's also important to note that for genes on autosomal chromosomes, these are the normal chromosomes, the non-sex chromosomes, if you see the recessive phenotype, you're looking at a homozygotic recessive genotype. It just goes with the package of it. The only way an individual can show a recessive phenotype is if they are homozygous recessive, by definition. The only other allele an individual could have would be a dominant one, and if they have a dominant allele, they show the dominant phenotype. So anytime you're told you're looking at a recessive individual, you instantly know the genotype. The genotype is homozygous recessive. But back to this tall plant. So I have a tall plant. That's all I know about it. I know it's not short, but I don't know what its genotype is. With whom can I mate this tall plant to determine conclusively its genotype? That's the test cross. I would, of course, mate it with a short plant. A short plant has nothing to contribute to the next generation but short alleles. And as we said in the last lecture, if my tall plant is big T, homozygous dominant, then that tall plant has nothing to contribute to the next generation but dominant T alleles, and every single plant in the subsequent generation would be tall. So that's the prediction. I take my tall plant of an unknown genotype, I mate it with a short plant. If 100% of my baby plants are tall, then I know that my tall parent is homozygous tall. If half of my baby plants are tall and half of them are short, what brought that shortness out was a recessive allele in my tall parent, and so I know that the tall parent was indeed heterozygotic. That is the test cross. The textbook says, when a test cross is performed, any recessive allele in the unknown genotype, that's the genotype of the dominant expressing parent, is expressed in the progeny. I'll say it again. When a test cross is performed, any recessive allele in the unknown genotype is expressed in the progeny because it will be paired with a recessive allele that came from the homozygous recessive parent. That's the point. So given all that probability that we're now comfortable with, the definition of a test cross, which we're comfortable with, let's go ahead and do some 2-gene crosses and talk about what that works, how that works. We've talked about it a number of times, but the principle of segregation is Mendel's principle that parents have 2 genetic elements that they can pass on to a child, but they only pass on one, and the one that they pass on has an equal probability of being passed as the other. We tied that into meiosis in the last lecture and said that's the case because chromosomes separate during anaphase 1 of meiosis, and as they separate, they go to one gamete or the other. Imagine a cell, or a pea plant, an organism, that is heterozygotic, but it's heterozygotic at 2 different genes. So now the genes are the R gene and the Y gene, and if this cell is heterozygotic at both genes, it's big R, big Y, little y. This cell is going to undergo meiosis. So first the chromosomes replicate, giving rise to sister chromatids, and we have 2 identical copies of each sister chromatid. But as you see in this figure, the R gene and the Y gene are on completely different chromosomes, and so they're going to assort independently. They're going to segregate independently of one another. They're not tied to each other at all. This cell undergoes meiosis, so after anaphase 1, chromosomes are pulled to different cells, as we see down here. And then after anaphase 2, sister chromatids are separated into individual gametes, and we are left with gametes containing one of each allele. But they independently assorted, so every possible combination is possible, and every possible gamete will be made. What I mean by that is we can have a gamete that contains both dominant alleles, big R, big R, big Y. We can have a gamete that contains both recessive alleles, little r, little y. We can also make the gamete that contains one dominant R allele and the recessive Y allele, and we can finally make the gamete that contains one recessive R allele and one dominant Y. Independent assortment says as long as these genes are on different chromosomes, not only can you make each of these gametes, you will make them all, and you will make them in equal proportions. This is because of the way that chromosomes segregate during meiosis. Independent assortment. Let's imagine the same scenario. Now it's a plant. It's a heterozygous plant at 2 different genes. So it's big R, big Y, little y. We self-cross that plant. What type of progeny should we get, and what ratios would we expect to get that in? What we really mean is that each of these possible gametes can fuse with each of the other possible gametes. Again, it's a self-cross. So we can get the big R, big Y gamete fused with the big R, big Y gamete here, but this gamete can also fuse with this one. Let me draw that out for you, rather than skirt it. So we can get this gamete fusing with that, this gamete fusing with that, this gamete fusing with that, or this gamete fusing with that. But we can also get this gamete fusing with this gamete fusing with this one. So you get the point. Every and all gamete can be made. Every and all combination of fusion can occur. It seems to become a bit of a mathematical nightmare, but it's not. As a side note, it's important to note that segregation and independent assortment are almost the same thing, but not quite. Segregation simply says that during meiosis, chromosomes separate. Well, duh, we know that. We saw that. Segregation is that chromosomes are pulled apart to opposite sides of the cell during anaphase 1. One new gamete gets half of the original chromosomes, that's the reductive division, and the other gamete gets the other half. Independent assortment goes one step further and says that when that happens and chromosomes segregate, they do so independently. So that as long as genes are on different chromosomes, which gamete got one allele has no impact on whatsoever which gamete gets the other allele from the other gene. In other words, when I roll a 1, and then I roll that die a second time, the 1 I got in the first roll has no impact whatsoever on what I might get in the second. in the previous example on the previous slide, what R allele a particular gamete gets impacts not in any way, shape, or form what Y allele that same gamete gets. So one gamete doesn't get all of your mom's chromosomes, all of your mom's alleles, and the other gamete gets all of your dad's. Chromosomes get shuffled. When this concept is applied to heredity, it means that if we go back to that plant, we think of that plant with its R and Y genes, the R and Y genes are on different chromosomes. That means the R and Y alleles assort independently. So gametes come in all possible combinations, just as we just showed in the previous slide. You get both dominant alleles together, both recessive alleles together, and one of each. How about genes that are on the same chromosome? What happens then? Well, it's different, and it's a little bit more complicated still, and we will cover that, but we'll cover that in another few lectures. So for now, just worry about genes that are on different chromosomes that assort independently. So we're going to do this cross. We're going to self-cross this heterozygotic plant that is heterozygotic at two different genes. Can you do the Punnett square? Yeah, you could. It would look like this. It would be a 4 by 4 square. But you can see this is quite a bit more complicated than what you're used to seeing. Now imagine if you were to go to 3 genes. Well, then you'd be dealing with a 6 by 6 Punnett square, 4 genes, an 8 by 8 Punnett square. It begins to be overly complicated very, very quickly. So how else can we do this? How else can we do this problem? Especially since I'm not allowing you to use Punnett squares for dihybrid crosses. We're going to use those probability skills we honed at the beginning of the lecture. So we now have all the tools we need to understand and predict the matings that involve 2 or as many as we want unrelated, unlinked genes on different chromosomes. We'll use Mendel's example first. Mendel crossed pea plants that had round and yellow seeds, 2 different traits, seed shape and seed color. He started off with homozygous, pure breeding plants. We know what that means now. Homozygous across the board, members of strains. And he crossed those with homozygous, pure breeding, green, wrinkled seeded plants. This is the cross we're talking about. Homozygous round, homozygous yellow, dominant for both, crossed with homozygous green, homozygous wrinkled, recessive for both. We don't need to really do any math yet, right? If we see this, we can predict what the F1 is going to be. The F1 is going to be 100% round and yellow, and the F1 is going to be completely heterozygotic. This parent has nothing to contribute but a big R allele, and this parent has nothing to contribute but a little R allele. So that's going to be the only possible genotype in the F1 for seed shape. This parent has nothing to contribute but a big Y allele. This parent has nothing to contribute but a little Y allele. And so the only possible genotype for seed color in the F1 is a heterozygotic. Every single offspring in the F1 will be heterozygotic, but all of the offspring will be round and yellow because those are the dominant traits. That's easy. But now Mendel took those F1 progeny and he crossed them. He selfed them. So now we're looking at an heterozygotic by heterozygotic cross. And what do you think he saw? Well, your first instinct might be the 3-to-1 ratio. 75% of them round and yellow, 25% of them green and wrinkled. I would understand why you would go down that road, but that's not what he saw. It is a heterozygote by heterozygote cross, but those 3-to-1 ratios, those pertained to single gene crosses, and this is a dihybrid cross. So let's solve this problem in a way that makes some sense to us. Let's use our probability. When solving genetic problems using dihybrid crosses or above, trihybrid crosses, tetrahybrid crosses, anything more than one gene, you always want to break that problem down into its individual single gene problems, the individual monohybrid crosses that are occurring within the larger problem. And there, you can use Punnett squares. So a dihybrid cross is really nothing more than 2 monohybrid crosses occurring together. So we're going to take our dihybrid cross, which is this one, the heterozygote by the heterozygote, and break it down as 2 monohybrid crosses. First dealing with seed shape, and then dealing with seed color. And we're going to use this Punnett square as our general guide for Punnett squares till we can refer to it. See, both of these crosses are heterozygote by heterozygote, heterozygote by heterozygote, and this Punnett square is the same, heterozygote by heterozygote. So this is going to be our guide Punnett square. Let's work this problem. If a heterozygous round yellow-seeded plant mates with another heterozygous round yellow-seeded plant, what proportion of the offspring will have round and yellow seeds? It's the same problem we had on the previous slide, the one where we said, well, maybe we're thinking 3-to-1 because that's what we expect heterozygotes to do when they're selfed. So it's the same exact problem. What we got to do is break this down. We have to consider each of the genes separately. We have to consider this as single monohybrid crosses. So what proportion of the offspring will just have round seeds? Let's disregard seed color for a second. Well, round seeds is dominant. We're dealing with a heterozygotic cross. This is our little Punnett square that we can refer to. So when we take a heterozygote for round and mate it by a heterozygote for round, we get 3/4 of the progeny being dominant round. So 3/4 of the progeny will be round. So we remember that. 3/4. Now let's switch to seed color. What proportion will have yellow seeds? Well, it's the same question. It's a heterozygote by a heterozygote. Here, yellow is dominant, and so it's 3/4. 3/4 will be yellow. But here is the real kicker. 3/4 will be round, and 3/4 will be yellow. That doesn't answer the question. The question really asked, what proportion of the progeny will be round and yellow, and yellow, and yellow? A little voice in the back of your head should be whispering so sweetly, pleading with you, product rule, the product rule, please don't forget the product rule. And you shouldn't. The product rule says that when we have AND events, we take the individual probabilities of the individual events and we multiply them. Well, what's the probability of being round? 3/4. What's the probability of being yellow? 3/4. We just demonstrated that with Punnett squares separately. But this is an AND event. What's the probability of being round and yellow? 3/4 times 3/4, which is, remember, when you multiply fractions, you multiply the tops and you multiply the bottoms, 9/16. 3 times 3 is 9. 4 times 4 is 16. 9/16 of the progeny will have round yellow seeds. What we did was we calculated the probability of just the roundness, 3/4. We calculated the probability of just the yellowness, 3/4. And we calculated those individually by using Punnett squares, treating them as single gene crosses. And then we came back to the problem and said, what did we want to know? Round and yellow? So we want to take those two and multiply them. Pretty straightforward. Our fraction is out of 16 because we have 16 possible outcomes. We can do the same with any of the phenotypes we want to look at. What's the proportion of round green seeds? Well, think about that for a second. What's the proportion of round green seeds? Well, we already know round. Round is 3/4. And what about green? Well, we can go back and see. Here's green, right here. The probability of getting green in this cross is 1/4. So that's 3/4 round times 1/4 green. The question is asking about the probability of round and green seeds, and so that's going to be 3/16. How about wrinkled and yellow? Well, wrinkled is the recessive characteristic for shape. It has a 1/4 probability. Yellow we've already calculated. It's this 3/4 from here. And so the probability of being wrinkled 1/4 and yellow dominant is also 3/16. How about the two recessive traits? What's the probability of being wrinkled and green? 1/16. So the phenotypic ratios for a dihybrid cross that crosses two heterozygotes is 9 to 3 to 3 to 1. I'll say that again. The phenotypic ratio for a dihybrid cross where the two parents are heterozygotic for all the traits is 9 to 3 to 3 to 1. That's 9/16 dominant for both traits, 3/16 dominant for the first but recessive for the second, 3/16 recessive for the first but dominant for the second, and 1/16 recessive for both. If you were to do the Punnett square, that's exactly what you would see. This Punnett square comes out with 9 organisms, 9 seeds that are round and yellow, 3 seeds that are yellow but wrinkled, 3 seeds that are green but round, and only 1 lonely seed that is green and wrinkled. But the probability gets you there. So we had a dihybrid cross where a heterozygote for both traits mated with a heterozygote for both traits. Now I'm going to ask you a genotypic question. Previously, we were talking about phenotypes. What's the probability of getting the heterozygotic genotype for seed shape and the recessive genotype, homozygote recessive genotype, for seed color? Well, the first question is what's the probability of getting that heterozygotic genotype for the first gene? Remember, you always break it down into individual monohybrid crosses. Disregard seed color for a second. Just look at seed shape. Well, with that particular cross, with that particular Punnett square, if we cross a heterozygote by a heterozygote, we have a 1/2 chance of getting that heterozygotic genotype back. This is the cross that we're doing. We're just considering seed shape. We can fill in the Punnett square. This is our first heterozygotic right here. Here's our second heterozygote right here. And that's out of 4. So we have a 1/2 probability of getting a heterozygotic genotype, given the cross with just the R's. What's the probability of getting the recessive YY genotype? That's 1/4. Now what do we do? Well, the question is really asking, what's the probability of getting the heterozygote for R and the homozygotic recessive for Y? That's a 1/2 times 1/4, which is an 1/8. How about this? Try to do this with a Punnett square, and let me know when you get it 5 years from now. We're taking a cross of heterozygote for A, heterozygotic for B, homozygotic recessive C with a heterozygote, heterozygotic D with a homozygotic recessive, and then heterozygotic E, and we're crossing those two parents. And I'm going to ask you, what's the probability, given this mating, of getting an offspring that has the homozygotic recessive genotype across the board? You take a look at this and you say, oh man, is he crazy? Well, we've been talking about this for an hour, not even, and he expects me to do this problem? But you can. Calm down, take a deep breath, and break it down into the individual monohybrid crosses. Just the A's first, just the A's. If you just consider the A's, what's the probability of getting the homozygotic recessive A genotype with just the A's? It's 1/4. When you take a heterozygote and you cross it with a heterozygote, you have a 1/4 chance of getting the 2 recessive alleles. Now just the B's. Given these B crosses, what's the probability of getting the homozygotic recessive B's? Same thing. It's 1/4, because for the B's it's also heterozygotic by heterozygote. Now the C's. What's the probability of getting 2 recessive C alleles given this mating of C's? This is a little different because it's a homozygotic recessive in the parent crossed with the heterozygote. So if you did that kind of quickie Punnett square on a side piece of paper or in your head, you would get a 1/2 probability for that. Now, what about the D's? The D's are also a 1/2 because it's a heterozygote by a homozygote recessive. And the E's are 1/4 because it's a heterozygote by a heterozygote. These are simple monohybrid crosses here that we've just broken down one at a time. Now we just have to remember our rules of probability and realize that the question is really asking us, what's the probability of being homozygotic for A and homozygotic for B and homozygotic for C and for D and for E? This is the product rule. So we take these individual probabilities and we multiply them. 1/4 times 1/4 times 1/2 times 1/2 times 1/4 is 1/256. And that's it. You could never do this so quickly with a Punnett square. Never. And look how quickly we've done it together in such a short amount of time. Just break these large multi-gene crosses down into their individual monohybrid crosses and recombine them with the product rule. Easy peasy bumpin' easy. All of this works because these genes assort independently. They are truly, probabilistically, independent events that just happen to be occurring together because that's the way we're considering them. Your textbook in this chapter goes through an alternative method for solving dihybrid cross problems. They're called branch diagrams. This is an example of a branch diagram. I'm going to be okay with branch diagrams. I've heard in the past that some high schools teach genetics with branch diagrams. I think that this method is almost as bad and almost as cumbersome as Punnett squares, especially when you get to the higher number of genes, like 3, 4, 5 gene crosses, the one we just did. But if you want to use branch diagrams, I'm not going to stop you. I just really think you're wasting your time by doing so. Branch diagrams certainly don't simplify the kind of problems we just went over with 5 genes. If you are interested in branch diagrams, but you haven't seen them before and it's something you want to explore further, come see me in office hours if you'd like, and I can explain them to you more one-on-one, but I'm not going to take the time here simply because I think that they're an inferior alternative. Alright, so now we're going to take our knowledge of dihybrid crosses and our knowledge of test crosses and combine it. Hopefully you remember from the last lecture and this one that a test cross is when we take a dominant phenotypic individual of an unknown genotype and cross that individual with a recessive individual of known genotype. Of course, the genotype is known. The only way the individual is recessive is if they have recessive alleles. So we have an organism that shows a dominant trait, or traits, because we're dealing with dihybrid crosses. We don't know if that individual is homozygous dominant or heterozygotic, and we cross it with a homozygotic recessive organism that shows the homozygotic recessive trait, and we observe the progeny. Remember, the basic rule of thumb is if we see recessive traits in the progeny, the dominant parent had to have contributed a recessive allele. The dominant parent had to have been heterozygotic. If we don't see that recessive trait in the offspring, chances are it's because the dominant parent had nothing but dominant alleles to contribute, meaning that the dominant parent was homozygotic dominant. If all the kids are dominant, chances are high that the dominant parent was homozygotic. We're using the same logic here, but we're applying it to dihybrid crosses. And sometimes we can make our lives really, really easy. Sometimes we don't have to do math at all. Here's our test cross. This is how we dictate or picture test crosses. This is really telling us that we have a dominant individual, an individual that in this case has round yellow seeds, and we're crossing it with an individual that has wrinkled green seeds. Now, the wrinkled green seeds, we know the genotype. The only way you have wrinkled seeds is if you're homozygotic recessive, and the only way you have green seeds is if you're homozygotic recessive. And so it's easy to tell the genotype of the recessive individual. That's the point of the test cross. What this denoting shows us is that we know that the individual must have at least one dominant allele because they're showing the dominant trait, but we really don't know what goes here. Do they have 2 dominant alleles, or do they have 1 dominant allele and 1 recessive? We don't know. That's the point of the test cross. And please keep in mind that it's not tied together. The individual can certainly be homozygotic for the seed color and heterozygotic for seed shape. There's nothing saying that the genotypes have to be the same in both of those genes. So given this test cross, the proportions of the progeny wind up being 1/4 of them round and yellow, 1/4 of them wrinkled and yellow, 1/4 of them round and green, 1/4 of them wrinkled and green. Based on this, do you know the genotype of the dominant parent? Here's one of those cases where you really don't have to do any math. If this parent were homozygotic dominant, every single one of the offspring should be round, because this parent would only have dominant round alleles to contribute. But look, we see that half of the offspring are wrinkled. Disregarding seed color, half of the offspring are wrinkled. So if half of the offspring are wrinkled, then most certainly we had to have had a recessive allele here in this parent. And then it makes sense that half of the offspring are wrinkled, because if we predict taking our heterozygotic parent and mating it with our homozygotic recessive, we would see half of those individuals showing the dominant trait and half of them showing the recessive. Now we consider seed color. If this parent were homozygotic dominant for Y, all of the seeds would be yellow, because this parent would have nothing but yellow alleles to transmit. But we see that half of the offspring actually have green seeds. If half of the offspring have green seeds, it's because this parent had a recessive green color allele to give to the next generation. And so without doing any math at all, by just inferring the genotypes of the parent, based on what we see in the offspring, we know that this parent must be heterozygotic at both traits because we see both recessive phenotypes come out in the offspring. Always think one gene at a time. Even if you're not doing probability and math, always consider the genes one at a time. It's really the only way to do it. And if you try to do it any other way, you're going to get yourself in a whole heap of trouble. It's just going to make things completely complicated for yourself. So here's a worked problem. This same problem that I'm about to show you is solved as a walkthrough in your textbook. So the textbook kind of breaks it down for you and walks you through it. And I am going to solve this problem for you at this website. So you can feel free to pause this slide as soon as I show the problem, kind of launch a new window in your browser, plug in this URL, and it's going to bring you to an online tutorial. I just wanted to do a lot of writing and a lot of breaking the problem down. You can see the writing with this thing isn't really so good on Tegrity, and so I'm going to redirect you to that website where I can really break the problem down for you. And if after watching my tutorial or reading the textbook, you still don't understand how that problem was solved, you still don't understand how to solve a problem just like that, you need to come see me immediately. And I stress this now. I'm not a scary person. I'm actually quite nice, especially to my students. Maybe not so nice to my kids, but to students, absolutely. I have patience, so please come to office hours if you're struggling. We're going to continue to build on this material. It's going to get more and more intense. So if you're struggling now, you need to resolve that so that you can build on a solid foundation for this material. I expect you all to master the sorts of problems I'm about to show before exam 1. Hint, hint, wink, wink. You can expect to see problems like this again, so be sure you're comfortable with it, because there are points to be earned or points to be lost if you're not. Here's the problem. The problem is we're dealing with mice. We're dealing with two genes in mice. We have coat color and coat pattern. We have black coat alleles, which are dominant over brown coat alleles, and solid patterns, meaning the coat is all one color, dominant over spotted patterns. The rest of the problem is read to you and broken down for you on that tutorial. So again, please pause the lecture now, go to that URL, and see how this problem should be solved. All right, so we're going to end up with this idea of a goodness to fit chi-square test, the statistical side of genetics. I want to reemphasize that I will not have you solve chi-square problems on any exam or quiz. I may ask you what the values mean, and I'll be a little bit more explicit about this as I go through the material. I might ask you what particular results of chi-test squares mean, what they represent, but I will not be asking you to do chi-square calculations yourself from scratch. I'm exposing you to this material so that it's familiar to you. I do not expect you to master this particular material. All right, so textbook goes into much greater detail for this. You're only responsible for what I teach. Remember, if I don't teach it to you, you're not responsible for it. So you should know these concepts. You'll cover them in statistics if you haven't already, but they're important for genetics, and I'd like to put these concepts into that genetics framework. So imagine I do a cross, some genetic cross. Let's say I cross a homozygote recessive with a heterozygote, and because that's the cross, I expect a 1-to-1 ratio. I expect there to be equal numbers of both phenotypes. So if I have 40 offspring from this cross, I would expect 20 to be of one phenotype and 20 to be of the other. But that's the predicted frequency. Let's say I actually do the cross and I get 18 showing the dominant phenotype and 22 showing the recessive. Well, it's close to 20/20. How many of you think that cross, quote-unquote, worked, that my outcome predicted my prediction, or matched my prediction? Well, it's close. Some of you might think they didn't. Some of you might be sticklers, want to see 20/20. It's debatable. Well, now let's say I do my 40 crosses, my 40 offspring, and I count them up, and 15 of them are dominant and 25 of them are recessive. Still think it worked? Still think my outcome matched my predictions? Starting to think maybe they didn't? Don't know. Starts to get a little bit gray. The goodness of fit test is a statistical test based on the chi-squared calculation that takes real-world experimental data, which is never, ever perfect, such as 18 and 22, 15 and 25, and tests it. And the test that's performed determines or tells the experimenter how likely is it that the numbers you see arose by chance randomly, in other words, have nothing to do with your prediction at all, as opposed to the numbers being what they are because of your prediction. In other words, the chi-square test tells you how trustworthy 18 and 22 are if your prediction was 20 and 20, and tells you how trustworthy your prediction of 15 and your outcome of 15 and 25 are, given your prediction of 20 and 20. This is the chi-square test in all of its glory. The chi-squared value is this value right here. This is what we're calculating. It's calculated by taking the sum of each of the individual observed values that you got from your experiment and subtracting from it your estimated values that you got from your prediction. You square each of those differences, divide it by the expected value, and sum those up for all of your data sets. Many of you might have said at some point in your life, I love science, but I hate math, so I can't do physics and I can't do chemistry. I'm going to be a bio major and stay away from math. And you say, well, you know, when it comes to genetics, no one can escape from math. Sorry about that, but it's just the way it is. Again, O is for observed. These are the values that your experiment spits out, like the 25 and 15 in the previous slide. E is for estimated or expected. These are the predicted values that your hypothesis predicts, like the 2020. You run your data through this formula, you get a value called chi-squared in the end. That's one of the two values you need for the goodness of fit test. The other value is easier to determine. It's called the degrees of freedom. What you do is you take the possible number of outcomes for the entire experiment. In this case, the possible number of outcomes is phenotypes, and that number is 2, the dominant or the recessive. We had no other alternatives. And you subtract from that number just 1. So 2 possible phenotypes, the degrees of freedom, subtract 1 from that and you get 1. If you had 17 possible phenotypes, you would still just subtract 1, and then your degrees of freedom would be 16. But you take your total number of possible outcomes and subtract 1 from it. You then use these two values, your degrees of freedom and your chi-squared value, on a chi-squared table, and you get a probability. Here's a chi-squared table. Let me get those markings out of there. Chi-square tables are standard. There's only one of them. You can find them in many, many different textbooks, many, many different resources. It's just a way to match your degrees of freedom with your chi-squared values, and it gives you a probability. So let's imagine your degrees of freedom were 1, as we said. So here's our degrees of freedom. And the chi-squared value you got was 1.7. So you would come to your row of degrees of freedom 1, and your chi-squared value is 1.7, so it's higher than this and lower than this, so your chi-squared value falls right in between these two ranges. And so your chi-squared prediction, the value of your test, the outcome of your goodness of fit test, would be somewhere between these two values, 0.25 and 0.1. That would be your result. And you'd be done. And you'd say, what the hell does that mean? Well, the outcome of the goodness of fit test is a probability. And the probability you're getting is the probability that the numbers or data that you obtained arose by chance. In other words, was random and had nothing to do with your prediction at all. That's bad. So the higher the probability value on this test, the worse your experimental data. A very high probability means it's extremely likely that the numbers you got were random and therefore had nothing to do with your hypothesis at all. A very low probability on this side of the table says that it's very unlikely that your data was random and arose by chance, and so very, very likely that your prediction was true. So you're looking or hoping for low goodness of fit data, goodness of fit results. So high chi-square values are bad because it says random chance explains your data. Low values for this type of test are good. It tells you that the data is real or the data was obtained in line with your prediction. So here's an example from your textbook. In this case, this mating is done. We don't really know the genotypes in this example, but we know the F2 generation gives us 105 purple plants and 45 white plants. Those are the observed values. The experimenter believes that this is due to the 3-to-1 ratio of Mendelian genetics. And so the experimenter says, well, if I had 150 progeny, I would expect, given Mendelian proportions, 3/4 of them to be purple, or 112.5, and 1/4 of them to be white, or 37.5. Those are the expected or estimated values for this test. The chi-square test then says, well, you take your observed, you subtract from it your expected, and you square it. And you do that as a sum for all of your data sets. So you do it for the purple, and you do it for the white. You blow through those numbers, and you generate a final chi-squared value. In this case, it's 2 with rounding and all of that. So 2 is your chi-squared value. You then have to determine your degrees of freedom. Your degrees of freedom are your total number of possible outcomes. 2, purple or white, subtract 1, and so 1 is your degree of freedom value. You then go to your chi-squared table with both of those values. So we had a value of 1 for our degrees of freedom and a value of 2 as our chi-squared value. So that falls right in this range here. I use a different table than was used in the textbook. You'll see what that means in just a second. But what this table is telling us is that the probability that the data arose by chance and has nothing to do with our prediction at all is somewhere between 0.25 and 0.1. Like I said, the table was a little bit different, so they have a larger range. Here they determine it's between 0.5 and 0.1. But close to what we're seeing. It depends on the table that you use. Is this good? That's for you to decide. Is it bad? That's for you to decide. I can tell you what this data says. This data says that it's between 10% and 50% likely that the data you found is due to chance. Between 10 and 50% likely that it's random chance. In other words, it's between 50 and 90% that your data arose because of your predictions, or in other words, your prediction is observed. You have to come to some consensus of whether or not that's a good number or not. So as a general rule, what we use as our cutoff for believable data is a chi-squared value of 0.05 or 5%. This is considered a significant or trustworthy data set, a significant or trustworthy result. This chi-squared value says, in other words, that there's a 5% chance or less that my data is what it is because of random chance. In other words, there's a 95% chance or more that my data is what it is because it matches my prediction. In other words, my hypothesis is correct. That's pretty good. Chi-squared values can get even higher, though. We consider a chi-squared value of 0.01 or less as highly significant or highly trustworthy. This means there's a 1% chance or less that I got my data simply by random chance. In other words, a 99% chance or more that my data is what it is because my hypothesis and prediction are correct. So highly significant or highly trustworthy. Always keep in mind, stats are stats. Kind of the quasiest math out there. I hate stats for many different reasons. Good chi-square values, good p-values pass the test of scientific significance, but they don't necessarily certify that an experiment was well planned out, was well carried out, was interpreted correctly. And remember, even though there's a 99% chance the data is what it is because the prediction is right, that still means there's a 1% chance it was random. And a 1% chance that it's random is a chance that it's random. So you've got to take all of this with a grain of salt. In addition, stats can be tweaked, and people can manipulate stats in technically, mathematically legal ways that are still manipulation, but I'll leave all that discussion there. No need to go into a rant about the failings and flaws of statistics. So what did we talk about today? Well, we covered a pretty wide gamut of stuff. We started off with a very kind of review, almost high schoolish introduction to probability, just to make sure we were all comfortable with that. A probability is the number of times an event can happen, divided by the total number of opportunities that event has in which to happen. We talked about solving genetic problems using probability, not using Punnett squares. We have the sum rule, which is given by the word or. The probability of flipping a head or a tail is 1/1/2 plus a half. And the product rule, and the probability of flipping a head and a tail is 1/4, 1/2 times 1/2. We went back to the idea of a test cross and kind of refortified the idea that it's used to determine the genotype of a dominant parent who has an unknown genotype. You cross that dominant parent with a recessive one. If you see recessive progeny, you know that there was a recessive allele in the dominant parent, making them heterozygotic. We use the example of a cell being heterozygotic for the R gene as well as the Y gene. Undergoing meiosis, chromosomes replicate, there's segregation and independent assortment, and so we get every possible genotype we can imagine. Big R with big Y, little r with little y, big R with little r with big Y. That led us into the conversation of, well, what happens if we cross organisms such as that? The so-called heterozygotic dihybrid cross. We do a dihybrid cross with an individual that is heterozygotic for both genes. And we see, using our rules of probability, that 9 to 3 to 3 to 1 ratio. 9/16 of the progeny will be dominant for both traits, R and Y. 3 out of 16 will be dominant for I but recessive for Y. Dominant for R but recessive for Y. 3 out of 16 will be recessive for R but dominant for Y. And 1 out of 16 will be recessive for both R and Y. We took the probability one step further and showed how versatile it really is. We did a 5-gene cross, given a known set of parents, and we predicted the probability of getting a specific genotype in the offspring. It'd be very, very hard to do with Punnett squares. And we stopped at 5 just because of considerations of time. Once you master this probability approach, you could do 12 gene crosses if you wanted to. It just takes a little bit more time to do the individual calculations. Remember the general rule of thumb, though. Always break down these large multi-gene crosses to their single monohybrid crosses. Consider them one gene at a time. Just the A's, then just the B's, then just the C's, and at the very end, combine those individual probabilities in an appropriate way, multiply them or add them as you see fit given the product rule or the sum rule. We finished with the goodness of fit test just to kind of introduce you as to what that means. I'll never give you observed data and expected data and ask you to give me a probability. I just want you to know what the chi-square test does, what it tells you, what the significance levels are, just the information that I presented. You don't have to do the calculations yourself. This test calculates the probability that our data arose by chance, randomly, and so the higher that probability, the worse our data set, because it means our data is pretty much random. The lower the probability, that means the less that probably arose by chance, or the more it arose because we're right. We consider a chi-squared value less than 5% as significant and less than 1% as highly significant. The next 2 lectures are going to take some of these Mendelian genetic rules and turn them on their head a little bit. The basic underlying biological principles will be exactly the same, but because of how life works and how chromosomes behave, we're going to have some exceptions to these rules. The next lecture will be on sex-linked traits. Those are genes and traits that are encoded on the sex chromosomes, the X and Y chromosomes, and then the lecture after that goes one step further and kind of extends and modifies some of Mendel's laws in ways that we wouldn't fully anticipate. But I'll save that for the next lecture. As always, thanks for watching, and we'll cover this stuff in class.